GARD platform can be viewed as a modular testing strategy framework based on interchangeable modular components. These components are the biological cell system and a training data set used both for biomarker discovery and subsequent training and definition of the prediction model. And then you have the genomic biomarker signature, which is specific for each assay and related to the mechanistic events associated with the individual toxicological endpoint being investigated. And then lastly, you have the prediction model, which performs the final classification and interpretation of the high dimensional data generated in the assay. So if we start with the biological cell system, it's of course helpful to know at least something about the mechanistic background of the toxicological endpoint you're investigating. And to this end, the AOP, or the adverse outcome pathway, may be of some assistance, illustrated in this figure. So this is quite common knowledge today, and much have been learned about the process of sensitization within the last 10 to 20 years. But in a brief summary, sensitizing chemicals penetrate skin and form protein-hapten complexes due to their reactive profiles in contact with proteins. These hapten proteins are taken up by dendritic cells, which mature and migrate to the local lymph node, where antigen presentation occurs to primarily specific T cells. So once primed T cells are available in a sensitized individual, upon secondary exposure, you will have a very rapid elicitation of the allergic responses. So the GARD platform utilizes the decision-making and the attenuation and initiation of downstream responses of the dendritic cells. And if we look at the dendritic cells, we today know that these cells react in a multitude of different ways in contact with these types of chemicals. You have recognition of foreign substances through innate immune receptors. You may have activation of self-defense mechanisms, such as upregulation of co-stimulatory molecules, including CD86. You may have cellular stress responses, as well as metabolic and oxidative stress responses, and of course, communication with different parts of the cell system, including chemotaxic receptors and antigen presentation functionalities of these cells. And all of these functions are, of course, controlled by the proteome of the cells, which is in turn regulated by the transcription from DNA to RNA. So this is the basis and one of the founding reasons that why we believe that transcriptomics is a very efficient way to get a holistic view of everything that's happening within these cells in response to chemical exposure. So with this knowledge in mind, the underlying hypothesis for each GARD assay is basically that there should exist genes and pathways in dendritic cells that are differentially expressed depending on the stimuli, whether or not it's a sensitizer or a non-sensitizer. And if we can find those genes, they can be used as predictive tools. So in order to test this hypothesis, each method development also starts with the definition of a training data set, or the creation of a training data set. And basically what we need to do is to identify a training reference set of chemicals with known properties relating to the investigated toxicological endpoint. So as an example, you can see on the right here an outtake of the training data set for GARD skin method. And having access to this training reference set of chemicals, we can create whole genome data, which can be mined for further information relating to the endpoint being investigated. Which leads me to the identification of a genomic biomarker signature. So having access to whole genome data generated from this reference set of chemicals, we can use statistical and computational methods to mine this data set in order to identify the genes that are differentially regulated in the two different groups of reference chemicals. And having access to a specific genomic biomarker signature, we are also able to train a computational prediction model, primarily using support vector machines that can assist in the interpretation of the high informational and high dimensional data that is being generated within each GARD run. So having all of these different modular components in place, we can define and freeze a finalized assay. And once we get to that stage, every GARD assay more or less follow the same protocol, which can be summarized in the following 6 steps. So firstly, we identify a suitable concentration in which we can expose the cell cultures of the test item. We do so by monitoring the cytotoxic effects induced by the chemical. Once we know what concentration to use, we can repeat exposure experiments in 3 biological replicates, and from these samples we can extract the genetic material or the RNA from these samples. Having access to the RNA, we can then perform gene expression profiling, and then we can actually quantify the expression levels of each of the biomarkers of interest for each individual GARD assay. Having access to the high dimensional data, we can feed this into our web-based applications, which holds the prediction models and the computational algorithm with output a classification stating whether or not it's a sensitizer or a non-sensitizer.